Hi, everyone. Happy Friday. Welcome to the Myotonic Dystrophy Foundation's Meet the DM Drug Developer Series. Today is April 1st, 2022, and we are talking with Dine Therapeutics. Just a bit about the Myotonic Dystrophy Foundation. Our vision is that we envision a world with treatments and a cure for myotonic dystrophy. The mission of MDF is community, care, and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care. We accelerate research towards treatments and a cure. Just a reminder of some of the support resources we have on our website, toolkits and publications at myotonic.org/toolkits-publications. Many of our support programs and information can be found at myotonic.org/find-support. Our calendar of events, which includes all of our support groups, as well as our conference and gala, and all of the activities that will be held by the foundation, myotonic.org/calendar/month. And our digital academy, which will include today's presentation once we have that recorded and up, as well as past conference and other presentations. That's myotonic.org/digital-academy. Well, today we are seeing Dine Therapeutics and hearing about their progress during this Meet the DM Drug Developer series. But you can join us every first Friday of the month at 12 o'clock Pacific. We are still waiting to hear from a couple folks for May and June, but we will definitely be holding the next, we will hold one on July 1st with Avidity Biosciences and August 5th with PepGen. Many of our drug developers and our industry partners will be at our conference in San Diego. So don't forget September 9th and 10th, San Diego, California. You can register in advance and submit your questions, even if you can't attend at 12 o'clock on the first Friday of the month at myotonic.org/meet-dm-drug-developers. We have many new upcoming Ask the Expert sessions on the third Friday of each month at 12 o'clock Pacific. Later this month, we will be hearing about key neurological features of DM1. In May, it'll be about primary care and DM. June 17th will be a little bit more than what the cost of DM might be. We'll be looking at really a community profile of the myotonic dystrophy community, looking at surveys and data that have been gathered. July, we're going to be talking about palliative care. August 19th, disability rights, future planning and special needs. And again, September 9th and 10th, San Diego. Register for any of these and submit your questions in advance at myotonic.org/ask-expert-series. Okay, you are really here to meet with Dr. Ashish Dugar from Dine Therapeutics. Dr. Dugar has over 20 years of broad experience across medical affairs, clinical and commercial development, and real-world evidence generation. Prior to joining Dine, Dr. Dugar served as Vice President and Global Head of Medical Affairs at Sarepta Therapeutics, where he built the company's medical affairs organization. Previously, Dr. Dugar led all commercial and market access efforts for intracellular therapies as Vice President and Head of Commercial Development. Dr. Dugar has also held executive management roles at Roche and Pfizer, and he began his career at the National Institutes of Health. He holds a PhD in pharmacology from Penn State College of Medicine. Okay, if today's program speaks to you, please consider showing your appreciation with a donation to MDF. It's myotonic.org/donate. And really, you are here to see, and we very much welcome Dr. Dugar. Thank you.
Great, thank you, Tanya. Good afternoon, everybody. I'm Ashish Dugar, the Senior Vice President, Global Head of Medical Affairs at Dine Therapeutics. I'm also joined on the phone by Molly White, who I think this community knows very, very well. She's the Head of Patient Advocacy at Dine Therapeutics. So I really appreciate the great introduction by Tanya. And I want to thank the Myotonic Dystrophy Foundation and Tanya and the organizers for inviting me to speak on behalf of Dyne. It's always a privilege to speak to the DM community and have the opportunity to share our progress. And today I'll talk about our FORCE platform and data from our Myotonic Dystrophy Type 1 program and our plans for entering the clinic. As Tanya mentioned, I've really tried to incorporate many of the questions that were already submitted. There are some that I won't be able to really address at this time, but I welcome the opportunity to come back here repeatedly to give updates to the community. So this slide indicates that we will be making forward-looking statements. Please refer to our SEC filing for more information. So at Dyne, it all starts with our mission. We're laser focused on delivering life-transforming therapies to patients with serious muscle diseases, with DM1 being a priority. Our mission is core to who we are and what we do every day. Over the past few years, we've been validating our scientific platform and technology, including our ability to target delivery of potential therapeutics to muscle. And we've generated very exciting preclinical data across multiple indications and disease models. And my focus today is on sharing our comprehensive data from the DM1 program and providing visibility into our plans going into the clinic. We've built a robust portfolio on rare muscle diseases. DM1 and FSHD, shown here, represent opportunities to develop first-in-class therapies. And in Duchenne, we're building a global franchise, starting with our Exon 51 program. I always like to spend a little bit of time on this slide as we're building Dyne to be the leading muscle disease company to deliver for families living with DM1 and other muscle diseases. We focus on 4 core strengths to help us deliver on that. First, we intentionally designed our FORCE platform to overcome the current limitations of muscle tissue delivery with modern oligonucleotides. The goal for our DM1 program is to develop a therapy that will lead to a reduction in toxic nuclear DMPK, correct splicing defects, and enable relatively infrequent dosing to potentially stop or reverse disease progression. Second is our focus on rare muscle diseases. We're advancing programs in multiple muscle conditions, and we've generated very exciting preclinical data that shows the power of our FORCE platform across diseases and in DM1 in particular. And I'll highlight some of that key data in this presentation. Our third core strength is our commitment to delivering for patients. We will leverage our muscle disease focus to advance our programs as fast as we can to the clinic. We're driving 3 programs to the clinic and plan to be dosing in the middle of 2022 for 2 of those programs, including myotonic dystrophy type 1. And finally, we have exceptional in-house and advisory expertise with deep knowledge of DM1 and novel therapeutics and a world-class scientific advisory board, which includes luminaries such as Dr. Charles Thorne. I'm definitely aware that you all know this condition very, very well. You know that DM1 is an autosomal dominant, progressive, multi-system, degenerative neuromuscular disease, most commonly diagnosed in adulthood. DM1 is caused by a high number of repeats of the CTG sequence in the DMPK gene. And these mutations cause toxic DMPK RNA to get stuck in the nucleus and bind with proteins involved in RNA splicing. This leads to abnormal gene splicing, which causes the symptoms of DM1. And DM1 can be categorized into multiple clinical phenotypes based upon the disease onset and severity. The most frequent phenotype of DM1 is the classical or adult phenotype. It's characterized by onset of muscle myotonia, followed by weakness and atrophy of skeletal muscles. Importantly, it's also characterized by cardiac conduction abnormalities and respiratory distress. And a big component of DM1, as you know, are the gastrointestinal and metabolic disturbances, fatigue, and sleep issues, which are all hugely impactful in terms of quality of life. So currently, no disease-modifying treatments are available, which limits, there's really limited treatment to manage the symptoms of DM1. To treat the genetic basis of disease, the key is to knock down nuclear toxic DMPK, which can then correct the splicing defect, which is why we call DM1 a spliceopathy. Now, Dyne's precision medicine approach is focused on changing the way therapy is delivered so that we can make a meaningful difference to patients. In our preclinical data, which I'll be going through in just a moment, we've demonstrated targeted delivery of Dyne 101, our clinical candidate, to skeletal, respiratory, and cardiac muscle, showing DMPK knockdown, reduction of foci, and correction of splicing, which is all critical to potentially modify disease progression. Here, I'll take a couple of minutes to describe our scientific platform, the FORCE platform. This drives our efforts to develop and deliver muscle-targeted therapeutics. It's expressly designed to address one major issue in muscle diseases, which is efficiently and reliably delivering antisense oligonucleotides that target the genetic basis of diseases so that we can address the issues in skeletal, cardiac, and smooth muscle. As you can see, Dyne's FORCE platform consists of 3 parts: a proprietary antigen binding fragment, or what we call a Fab, to enable targeted delivery of the ASO, the antisense oligonucleotide, to muscle, a clinically validated linker, and an antisense oligonucleotide selected to target the genetic basis of the disease. And on this slide, you can see this Y-shaped structure at the bottom right, which is a monoclonal antibody. So we chose a portion of this, our Fab, as I mentioned earlier, because we believe it offers powerful advantages over the larger monoclonal antibodies, including its smaller size, lower protein load, and the potential for better tolerability profile. The goal is ultimately to enable targeted, effective delivery of therapeutic molecules to skeletal, cardiac, and smooth muscle. Here on the bottom right, you can see that we selected the Val-Cit linker for our platform. This linker is what connects the Fab to the antisense oligo, which I'll talk about in the next slide. And we chose this Val-Cit linker because it's stable, it's been clinically validated, meaning it's been used in products that are on the market, so it's clinically validated in terms of safety and efficacy, and we believe this further de-risks our FORCE platform. So in the FORCE platform, the Fab and the linker remain constant. It's the antisense oligonucleotide that we can interchange depending on what we're trying to target. So the versatility and modularity of our scientific platform provides that flexibility to deploy different types of antisense oligos or other agents that are rationally selected to match the target biology for each disease. Now, where disease-modifying nuclear knockdown is key, as is the case for DM1, it's critical that we select an antisense oligo that goes to the nucleus. For this reason, we designed a proprietary ASO that targets toxic DMPK in the nucleus of the cell. The mechanism of FORCE is different from other approaches. We're essentially using the cell's natural biology to deliver our therapeutic to muscle cells. Our Fab binds tightly to the transferrin receptor type 1, or the TFR1. Now, TFR1 is highly expressed across the surface of muscle cells, and you can see it on the right-hand side of the slide here. And it's highly expressed across skeletal muscle, including diaphragm, cardiac muscle, and smooth muscle. So we're able to harness what already exists on the surface of muscle cells to deliver our therapeutic with this receptor-mediated mechanism. And by targeting TFR1, we can modulate the kinetics, we have a wide therapeutic index, and should be able to dose relatively infrequently when we get into the clinic. This slide goes into some detail on how FORCE targets toxic nuclear DMPK RNA. We're utilizing the natural cell biology, as I said before, of TFR1 receptor-mediated delivery to transport therapeutics across the cell membrane without interfering with the natural function of TFR1. Here you can see that the FORCE conjugate attaches to TFR1 on the muscle cell membrane in the top left of the slide. Once across the cell membrane, an endosome forms in the cell which contains our therapeutic agent. In the endosome, the ASO is cleaved from the Fab and escapes the endosome without the addition of any membrane destabilizing agents. The ASO then traffics to the nucleus, where it binds the DMPK RNA. Now, it's the toxic DMPK mRNA in the nucleus that binds these factors called splicing factors, like MBNL, shown here near the bottom of the slide, a muscle-blind protein. And it sort of captures these factors, and then they can't do their normal job. So our agent can help release those factors and restore their ability to function normally, and specifically to restore splicing activity. So I have a short video to demonstrate the mechanism by which the FORCE platform works to address the genetic basis of muscle diseases.
At Dyne, we are focused on advancing life-transforming therapies for people living with serious muscle diseases. We are utilizing our FORCE platform to develop modern oligonucleotide therapies that are designed to overcome limitations in delivery to muscle tissue. Our therapeutic candidates consist of three components: an antigen-binding fragment antibody, or Fab, a clinically validated linker, and an oligonucleotide payload. The Fab is conjugated or connected to the therapeutic payload using a linker. Our proprietary Fabs are engineered to bind to the transferrin-1 receptor, or TFR1, which is highly expressed on the surface of muscle cells. The Fab engages with the TFR1 to facilitate the delivery of the payload into the muscle, where it aims to target the genetic basis of disease. Dyne is advancing therapeutic candidates for myotonic dystrophy type 1, Duchenne muscular dystrophy, and facioscapulohumeral muscular dystrophy. DM1 is a rare progressive muscle disease caused by abnormal expansion in the region of the DMPK gene. This genetic defect leads to the production of RNA with high numbers of toxic CUG triplet repeats that form large hairpin loops and remain trapped in the nucleus. This toxic RNA sequesters MBNL splicing factors and causes abnormal splicing events, leading to the clinical presentation of DM1. People living with DM1 experience a wide range of symptoms, including progressive muscle weakness, myotonia, cardiac and respiratory problems, and cognitive impairment. We use a single-stranded antisense oligonucleotide, or ASO, which travels to the nucleus to target the toxic RNA and release splicing proteins, enabling normal RNA processing of many genes and potentially stopping or reversing disease progression. In a preclinical study, Dyne's lead DM1 candidate demonstrated significant reduction of toxic human nuclear DMPK RNA in multiple muscles. The modularity of the FORCE platform enables the selection of different types of payloads to match the genetic basis of the disease, including phosphorodiamidate morpholino oligomers, or PMOs, to promote nuclear exon skipping in DMD, and small interfering RNA for targets in the cytoplasm. With the FORCE platform, Dyne is aiming to deliver therapies with the potential to transform the futures of people living with serious muscle diseases.
Awesome, thank you. So hopefully the video was helpful to everyone. So all of you know DM1, of course, and the point I'll make here is our work is so important because we understand, we've heard from you what a devastating disease this is. And currently there are no approved treatment options for people like Joachim, who's pictured here. And so it is our mission to make sure that we're able to develop disease-modifying drugs for myotonic dystrophy. To prepare for our entry into the clinic, Dyne has deliberately conducted comprehensive preclinical studies across all of our programs. And we've not shied away from asking the tough questions or running the right studies to evaluate our product candidates. We've operated with a strong desire to understand our molecules and best position them for success for patients. The team has generated the most comprehensive, significant, robust data set across our programs that we believe sets a new bar in our field. Whether this includes experiments in patient cells, seen at the bottom of the slide, various mouse models, which are the next 3 rows moving from bottom to top, to help us understand the pharmacokinetics and distribution in muscle, the pharmacodynamics, meaning the DMPK knockdown, the splicing improvement, or even looking at function in the mouse model to help us understand effects on myotonia. And then we've also done studies in non-human primates or monkeys to understand if it's getting into muscles in larger species and how safe and tolerable it is. And I'll take you through a lot of this data over the next several slides. So I'll take you through the data on Dyne 101, which is our clinical candidate, in patient cell lines with approximately 380 and approximately 2,600 repeats. As you can see, these different cell lines represent a range of the patient population. And we're focused, our initial focus is on addressing the approximately 75% of patients who are either adult or late onset. So turning to the data on this slide, we showed a robust, dose-dependent DMPK RNA knockdown shown on the left-hand side of the slide in these histograms, and reduction in nuclear foci as represented by the reduction in the red foci or red circles, red dots in the panel in the middle. This resulted in correction of splicing defects as measured by BIN1 exon 11 inclusion. The results in the cell line with 2,600 repeats are particularly notable given the severity of DM1 disease represented by this high number of repeats. So again, we're able to show the knockdown, the foci reduction, and the correction in splicing in the patient cells. It's probably a story you'll hear over and over in my presentation with different models. The HSA-LR model, developed out of Dr. Thornton's lab, has CUG repeats in the nucleus, and it mimics RNA missplicing, and you've heard me talk about the missplicing, that's observed in patients. And it's really one of the very few models that presents with a myotonia phenotype, which is also seen in patients. As shown on the left panel, we observed dose-dependent correction of splicing in multiple muscles, including the quads, the gastroc, and the TA. Now, each of these points is a composite splice score of over 30 RNAs, including various channels like the chloride channel and calcium channel and other structural and functional proteins, demonstrating that we're normalizing multiple muscle proteins in this model with a single low dose of force given intravenously or via IV. On the right side of the slide, you can see the profound splicing correction with a single low dose resulted in unparalleled decrease in myotonia observed in this same mouse model. These studies were conducted blinded at a third party, and they demonstrated that a single dose of force nearly reversed myotonia in 3 separate muscle groups. To go into a little more detail, on the right-hand side, the saline treated animals in the black bars did not alter myotonia. In the red bars is what we call naked or unconjugated antisense oligonucleotide, which decreased myotonia slightly. And then our conjugated ASO, our force platform in blue, showed near reversal of myotonia with that single dose measured 14 days post-administration. This gives us confidence that we can deliver low doses of modern oligonucleotides infrequently into muscle to correct splicing and clinical phenotype. The literature indicates that the toxic human DMPK RNA resides in the nucleus and is the genetic basis of DM1. We set out to conduct our own experiments to understand this further, as well as the ability of Dyne 101, our clinical candidate, to reduce this toxic DMPK. So we developed the HTFR DMS-XL mouse model shown on this slide by crossing mice with the knocked-in human TFR1 receptor and the DMS-XL mice that carry the human mutant DMPK transgene with about 1,000 repeats. So this model expresses both the human transferrin receptor and toxic human DMPK, which allows us to test our clinical candidate. It helps with the translatability of what we see here into humans. We evaluated the impact of Dyne 101 in driving toxic DMPK knockdown in skeletal and cardiac muscles in this hemizygous mouse model. What I mean by that is they have one copy of the DMPK gene, hemizygous mouse. By way of background, multiple genetic studies in the field have indicated that aiming to target 30 to 50% knockdown of mutant DMPK has the potential to be disease modifying. We were pleased that after a single dose of 10 milligrams per kilogram, assessed out to 28 days, Dyne 101 demonstrated significant toxic human nuclear DMPK knockdown, about 46% in the heart, 51% in the diaphragm, 42% in the gastroc, and 46% in the TA. So we were very excited by the level of knockdown of the DMPK, of toxic DMPK, after just a single dose 4 weeks out. In this study, we wanted to confirm that toxic human DMPK is indeed trapped in the nucleus. So in the hemizygous model, as indicated by these fractionation studies where DMPK displays a similar expression to MALAT1. So you can see here on the left-hand side, the human mutant DMPK in the nucleus versus the cytoplasm looks similar to what you're seeing on the right-hand side in the MALAT1 histogram. And that MALAT1 is a nuclear RNA marker that's commonly used in studies. So once again, this gave us confidence that that toxic DMPK resides in the nucleus. We then took this model a step further, and using this mouse model, we were able to show that with Dyne 101, there is a substantial decrease or knockdown of DMPK, as you can see here. So again, we're able to show it's enriched in the nucleus and that Dyne 101 can actually knock down DMPK in the nucleus. So in this particular next set of data, we evolved the work that we were doing and created a homozygous mouse model, which has 2 copies of the toxic human DMPK. When we evaluated Dyne 101 in the heart of the homozygous mouse, so two copies of the homozygous mouse, after just 2 doses of the relatively low dose of 10 milligrams per kilogram, we observed reduction in foci and toxic DMPK of about 49%. And the unique thing about this model, unlike the hemizygous mouse model, we can also look at splicing correction. And so this resulted in compelling splicing correction at 4 weeks. And we know that cardiac muscle has been difficult to access in muscle diseases, and this is critical to improving the condition of patients. On this slide, we show that Dyne 101, shown in blue, in the blue bar, demonstrated a reduction in toxic human DMPK across various skeletal muscles, including diaphragm, TA, and gastroc, ranging from 40 to almost 50%. Importantly, this resulted in corresponding robust splicing correction after 4 weeks across all of these muscles. So we're really excited by the data from the numerous studies in these mouse models, as we believe our work with these models sets a new standard for evaluating DMPK knockdown and splicing in DM1. Developing this model was critical because non-human primates are not a disease model, but there's still a lot of important information that we need to glean from going to a larger species like monkeys. So we utilize non-human primates to evaluate our ability to get into the muscles engage the target, namely DMPK, and assess safety and tolerability. In one of our NHP studies, we demonstrated that Dyne 101 robustly reduced wild-type DMPK in multiple skeletal, cardiac, and smooth muscles. These data demonstrated that we can deliver into larger species and engage the DMPK target. This is very important information that we're able to glean from this monkey study. We also evaluated non-human primates in a 13-week GLP tox study. As shown here, we were very pleased to see that Dyne 101 was safe and well tolerated after repeat dosing. We observed no dose-limiting toxicity and no changes in cardiac, respiratory function, liver or kidney function, weight changes. And so this data gives us great confidence to move into the clinic. The slide summarizes the data we've generated in our program. I won't go through all of it again. Essentially, we believe that we've designed Dyne 101 to offer a number of advantages as we develop this agent and move into the clinic. We are leveraging TFR1 to solve the challenge of delivery to muscle. We've selected an antisense oligonucleotide to match the genetic basis of myotonic dystrophy type 1 to drive disease modification. And with this targeted delivery, we have the potential for enhanced tolerability, a wide therapeutic index, and relatively infrequent dosing. As you can see here, we have multiple regulatory filings planned in the second quarter of this year, which I'll talk more about in just a moment. And we anticipate dosing patients in the middle of this year. We've done extensive work to advance Dyne 101 to the clinic. We've engaged with stakeholders around the world as part of this effort. This engagement has included advocacy organizations, patients, key opinion leaders from various disciplines, including neurology, cardiology, physical therapy, pulmonology, and other areas, to ensure that we have their crucial perspective on how to inform our efforts. These thought leaders, with their tremendous experience as clinicians and running clinical trials, have provided us with critical input on the overall design of a clinical study, biomarkers, endpoints, including splicing, myotonia, various strength and functional outcomes, as well as key safety considerations. They've also provided key learnings from natural history studies. We've also reached out broadly to advocacy leaders, patients, and caregivers, many of whom are healthcare providers themselves, to ensure that we have a patient-centric study design. And they provided advice in a number of areas, including clinical trial assessments, placebo and biopsy-related considerations, as well as clinic visit designs and decision-making insights for the selection of participating in a clinical trial. So patients, caregivers, clinicians, and regulators have all shaped our thinking for our proposed design, a study design for Dyne 101. As you can see, we're planning a global placebo-controlled multiple ascending dose study of Dyne 101 among individuals with symptomatic DM1. We're aiming to enroll about 40 to 50 adult patients, and the study will also have a long-term extension. As for endpoints, we plan to evaluate safety and tolerability, of course, as well as splicing, measuring muscle strength and function, including myotonia, ambulation, respiratory function, and other parameters. As I mentioned, we anticipate dosing patients in the middle of 2022. For our DM1 program specifically, as we look to execute on our global plans, we now anticipate submitting regulatory filings in the second quarter of this year in multiple countries to support initiating our clinical trial versus our previous intention to submit an IND in the first quarter of this year. We've continued to engage with members of the global DM community as we prepare to enter the clinic, and we're moving as quickly as possible. This is an exciting time at Dyne as we prepare to transition into becoming a clinical stage company with 2 co-lead programs entering the clinic and a third not too far behind, and the opportunity to bring forth to many other diseases. The team is focused on execution on all fronts as we seek to bring our potential therapies to patients as quickly as possible. The voice of the patient and the urgency in DM1 is front and center for us. And it's a critical part of understanding how best to design and optimize our clinical program with no approved therapies available now. And we know that patients are waiting for us and waiting for other companies to be able to develop therapeutics to alleviate this disease. These are some of the quotes that we've heard from individuals living with DM1, and this speaks to the disease burden that the community is facing. As I said earlier, it's a privilege to be talking to this community. I want to thank you on behalf of Dine for giving us your attention today, and we hope that we can come back and continue to provide updates for the community. We look forward to it. Thank you. So I have some questions that hopefully were incorporated in the presentation. And then I have a few questions here in front of me that were submitted prior or earlier before this presentation. So I'll just read a few of them and provide a few answers. I will also invite Molly White to join me and help me with some of the Q&A. So while Molly is joining, hi there, Molly.
Hello.
You know everybody. So one of the questions that we had was, when will you release trial design details? Inclusion, exclusion criteria, more specificity on the outcomes that you'll be collecting, the sites that you'll be going to, etc. So there isn't much more that I can share in terms of the details of our study design at this point. As we get closer into the clinic, we'll continue to release more details about the specifics of the trial design, the protocol, the sites, inclusion, exclusion criteria, etc. So I provided what information I'm able to at this time to give you a sense of the kind of study we're conducting, this first-in-human multiple ascending dose study, looking at safety and tolerability, as well as some pharmacodynamic markers like splicing, and functional outcomes and quality of life outcomes as well. Another question we had is, do you expect your treatment to improve outcomes across muscle groups? It's a great question. I'm sure I don't have to tell any of you that the disease there's a tremendous amount of heterogeneity in this disease. What we are able to tell from our non-clinical studies is that we can get into the muscles that are leading to the phenotype seen in DM1. We're able to get into the heart. We're able to get into the diaphragm. We're able to get into the skeletal muscle. We're able to show that we are knocking down the toxic human nuclear DMPK and correcting splicing in those muscles. In the one mouse model, we're able to actually show the reversal of the Myotonia phenotype. So we're confident that we're able to get into the cells that we need to get into, and that the drug in these non-clinical studies, it's acting the way that we think it should be acting. So there's a lot of heterogeneity, and our hope is certainly as we get into the clinic, we're able to show that it's safe, it's tolerable, that comes first, but that we can also improve functional outcomes and quality of life. And this is one study of a series of studies that, of course, we'll be conducting over time. We did get a question on how will the drug be administered and with what frequency. So we will be administering the drug via IV, so it's intravenous. And given the data that I've shown you today, we believe it'll be relatively infrequent dosing. We think it'll be no more frequent than once a month, and we're still determining what our path forward is going to be in terms of dose regimen, but it's we're really excited about what we're seeing. So those are some of the key questions that I pulled out from the list that I think I didn't directly address in the presentation. Molly, is there anything you want to add or any other questions you see coming through?
We have gotten some really good questions through the question function while you were presenting, so I can run some of those by
Sure.
you.
Sure.
Since you can answer anything.
Or you
The
can
question that I think we also got last year is, how is your platform design different from Avidity's?
Yeah, great question. So first of all, let me actually preface it by saying, you know, Molly and I and everybody in the community, we're so excited that there is so much effort and focus in this space now. We truly hope that every single therapy gets approved, has a great effect on patients, improves quality of life, gets approved, and that patients and families have access to drugs. It would be amazing. The more options, the better. So we really truly hope that everything that's in development makes it to market. And the other nice thing about this is that there are different approaches. It's good to have different approaches so that there's more shots on goal in terms of what actually works for patients and for families. In terms of the specific question with respect to Avidity, you know, one of the things that we're doing, as I said, is targeting. Well, we're using an antisense oligonucleotide, and the reason for that is because we are targeting the toxic DMPK that resides in the nucleus, which is the genetic basis of the disease. And you know, I'm not going to get into a compare and contrast in any level of detail, but Avidity's a payload is a bit different in terms of the way it works and how it's targeting the disease. So we really focus on the toxic DMPK in the nucleus, and we use a antigen binding fragment. We're using a piece of the monoclonal antibody because we feel that its smaller size, for example, will allow better penetration into muscle cells, that it doesn't have certain components of the monoclonal antibody, which should give it advantages in terms of its any sort of immunogenic effects. So we're excited about our approach of an antigen binding fragment that is attached to an antisense oligo that binds to the TFR1 receptor, which Avidity does as well, and targets the nucleus, the toxic DMPK that resides in the nucleus.
Perfect.
Do you want to add anything, Molly? Please do.
I don't want to add anything. I think that was well done. Great question here on dosing in humans based on mouse data. How are you determining the dosing regimen and the dose levels based on mouse data? Do you want to talk about some of the dose range finding studies?
Yeah. So
process.
Yeah, of course. So I think it's a great question. We can spend an hour, I think, on that question alone, or you know, a week on that question alone. It's fantastic. So there's a couple of different ways to answer it. First of all, what we're doing in the mouse studies as well as the monkey studies is constantly evaluating different doses and different dose regimens within those studies to be able to understand essentially the magnitude of effect, the durability of effect with different doses and different dose regimens. So we've looked, so for example, what I did not present was data that showed after 2 doses a week apart of 10 milligrams, we looked at DMPK knockdown 2 weeks later. And actually that looked very similar to the knockdown we saw after a single dose of 10 milligrams 4 weeks later, which is the data that I presented. And so that's really important information because it shows us, it gives us information insight into dose and durability. So we do a series of experiments like this, both in rodents and in monkeys, to give us a good understanding of how much drug is getting into the muscle tissue, into the blood and muscle, how it's translating to knockdown, how it's translating to splicing at different doses with different dose regimens, and of course, looking at safety and tolerability. So we take all of this information to then help us think about what we want to do in the clinic. And it's all of this data that helps us feel confident that we can actually have pretty infrequent dosing when we get into the clinic. And of course, more details on all of that will be coming in the near future. So that's not information I can provide right now. So that's that piece of it. And then there's a deeper scientific piece, which I certainly am probably not entirely qualified to go through here, around the kind of scaling when you do studies in mice and monkeys and how that scales into a human equivalent dose. So there's a lot of thought that goes into a lot of experiments and modeling that goes into how you think about dosing from animals to humans.
Perfect. There's a few questions in here on how to get involved in the study. So do you want to talk a little bit about a phase 1/2 versus a phase 3, maybe the different sections of the study?
Absolutely. So maybe at a high level, I think this community is very well versed in drug development. So I know that that's not needed. But what I would say is thinking around drug development in rare diseases is maybe a little different than a traditional approach. And so for this first-in-human study, we are very much focused, as is often the case, on safety and tolerability to make sure that we are providing patients doses and escalating doses in a safe manner, first and foremost. We also want to be able to glean information with respect to some of these other aspects that I mentioned, like splicing and function. And in this first-in-human study, it's not uncommon to have a patient population that is, kind of narrower. You try to create a bit more of a uniform patient population, because you want to be able to, in a thoughtful, stepwise approach, understand what your drug can do, in patients. Then you move into the later stage development, phase 3, if you will, and a series of phase 3 studies where you expand the patient population. It's still tightly controlled. You're still thoughtful about your inclusion and exclusion, but perhaps you expand your lens a bit to be able to make sure that you have studies that are even more generalizable to the patient population at large. And then you go on to phase 4 studies and real world evidence studies where you actually open it up even further, that lens or aperture even further. So you can really understand in sort of this natural setting what the drug, what the drug is able to do in terms of functional benefit and quality of life. Hopefully that gives a bit of a lens into how we think about things. Molly, do you want to add to that?
No, I think, I know from my last job that everybody wants to be involved in, most people want to be involved in a study, most people want to have access to potentially transformative therapies. And it can be really upsetting, and it can be really frustrating to feel like you don't understand in a transparent way how to access these studies. Why do some people get picked and other people not get picked? Is it fair? How are these decisions made? And I think, there's some information that can't be shared, but I think it's really important for people in the community to understand that the worst thing in the world would be if there was a potentially transformative therapy for myotonic dystrophy type 1, that because the patient cohort that was involved in the study was too broad, or the study wasn't designed effectively, despite the fact that the drug worked, the trial fails, and you have to go back to phase 1. You have to go back to, you have to start all over, and it's a multi-year process, as everybody on this call knows. So I think, while I really personally empathize with the frustration people feel when they don't understand why they are or aren't included in a study and they want to be included, I think the thing we can say with from the heart is that we want to get everybody in. We want this to work incredibly well. We want to change the lives of people that we personally know and care about quite a bit. And we will do everything we can to make sure we have the right study in place to demonstrate whether the drug is working or not, and then get it to everybody that we can possibly reach. Yeah. And we will do that in as transparent and communicative a way as we possibly can. So I think that's probably the best that we can say right now. But as we move forward and we get to the clinic, and as Ash said, we plan to begin dosing participants mid-year 2022, which is right around the corner. We will be communicating through our team, our teammates like the Myotonic Dystrophy Foundation as often as possible to keep you apprised of what we're doing. And we know how important that is. I would say in that general vein too, we got some questions around what about DM2? Which, as you know, is very close to our hearts. And what about crossing the blood-brain barrier? How do we start dealing with some of the cognitive issues? You want to speak to some of that?
Sure. Why don't I start, and then I'll ask you to add, please. So as I mentioned, we're initially focused on addressing the approximately 75 patients with adult or late onset. However, as Molly just alluded to, we're committed to addressing first the full spectrum of DM1 patients, including those with a CNS phenotype. We're certainly, we believe there's an opportunity to leverage our expertise to develop therapies that cross the blood-brain barrier and address the CNS symptoms. And so we are, actively involved in that kind of research. So we're moving forward, and we're committed to serving patients across DM1, and then and across DM. We recognize that there's huge unmet need in DM1, DM2, and so we are continuing to move our programs forward to be able to address DM patients. Molly, did you want to add anything?
Sorry, I'm also writing down questions.
I know, sorry. No, I think we're good. I think we covered it.
Yeah, okay, great. I mean, I think we want to serve every DM patient we can target.
We can, exactly.
And we're committed to doing that. Yep.
We have a couple minutes left, maybe one more question or two.
I'm just trying to pick one that's the most representative.
Sure. I think I've covered all of the ones I have here.
There's a question about why is there a clinical hold on the DM1 program? Maybe you want to just address the fact that there isn't a clinical hold on the DM1 program.
Oh, interesting. Yes. So there is no clinical hold on the DM1 program, as Molly just said. We do have a clinical hold for our Duchenne program, which we've publicly put out a press release and all that stuff. And the FDA wanted more clinical and non-clinical information from our IND package, and we have also stated that we'll be providing our complete response to that in the second quarter of this year. But no, there's no impact of that on the DM1 program. And so there's no, just to be clear, there's no clinical hold on the DM1 program.
I think you tackled most of it.
All right. I don't see any other questions on my list either. So I think we've covered everything.
Everything we can cover.
Everything we can cover.
Thank you.
So thank you very much. Thanks, Molly. Thank you, Tanya and the MDF for inviting us. We truly appreciate it. And thank you to all of you listening out there.
Thank you both. That was fantastic. It's an exciting time. And thank you in particular for being so thorough and addressing so many of the questions and breaking it down so it was very, very clear. That is well appreciated. We are looking forward to mid-2022 and partnering with you to announce whatever may come at that moment in time. And we're excited to share out any new progress that you might have made also at the conference. So I think we're hopeful to see you in September in San Diego as well.
You will.
Fantastic. All right. Well, thank you, everyone. Thanks for being with us today. And have a fantastic weekend. Take care.
Take care. Bye-bye. Good weekend.
Bye.